Hello, welcome to Datta Biology classes. Today we will discuss about photorespiration. We have completed respiration and then we have completed photosynthesis. Now we will discuss a little small chapter. Sometimes it comes in the exam known as photorespiration. So there are two words inserted in single. Photosynthesis plus respiration jointly forms photorespiration. Why we will read this? We will read because something is related to photo and something is related to respiration. What is that? In photosynthesis, what we have seen that 6 CO2 joins with RuBP with the help of enzyme rubisco, as we say. It is RuBP carboxylase. RuBP carboxylase. They produces an intermediate of 6 carbon compound, which we are not going into deep. Ribose bisphosphate is a 5 carbon compound, and here it is 1 carbon. So these joins to form a 6 carbon compound intermediate, which is unstable, and thereafter it produces 12 molecules of phosphoglyceric acid. We have come across. So to 12 phosphoglyceric acid along with 12 ATP and 12 NADPH, it forms 12 molecules of PGALD, phosphoglyceraldehyde. In short, somebody tells us phosphoglyceraldehyde 3-phosphate, somebody tells glyceraldehyde 3-phosphate, and we will call it PGAL. These are 3 carbon compound. Out of this 12 PGAL, we read it that 10 PGAL goes back to form 6 RuMP, ribulose monophosphate. Ribulose is a 5 carbon compound. So 5 6 are 30, and here we are taking 10. 10 3 are 30 because it contains 3 carbon each. So 3 times 30, we have taken 6 RuMP, and thereafter 6 RuMP produces Ru bisphosphate, that is another phosphate is coming. So for 6 ATP is used to produce 6 ADP. And here we are left with 2 PGAL, that is 3 plus 3, 6. These joins to form glucose. C6H12O6. This was in the Kelvin cycle. This is carbon fixation, this, so many things. Anyway, now this is Kelvin cycle. So Kelvin cycle, what it is doing? RuBP is the CO2 acceptor. So it accepts CO2 and it produces Pg. And the cycle moves on. Why we have taken 6? Because we have to produce glucose at least 1 molecule. So for our convenience, we have taken 6. That is out of 6, 2 compound, 2 PGALD was produced, and this 6 produces glucose. No problem. Another 10 we have taken to produce 6 RuBP. So the reaction is balanced. So now while we are reading this photosynthesis and respiration, this is known as Kelvin cycle. This is simply a recapitulation. Why we are not reading it? Because earlier it was thought that the rate of respiration was same in the light as well as in the dark. As we respire, we do respiration in the day and in the night. Plant also respire in the day and in the night. So earlier it was, they were in the view that yes, the rate of respiration is almost the same. But subsequently it was observed that the rate of respiration in the light proceeds at a higher rate. That is higher respiration, higher respiration, higher respiration. So respiration is more in the daytime than in the dark. So scientists are astonished that what happened actually, because in the light the respiration, rate of respiration increases than in the dark. In the dark, rate of respiration normalizes. So there must be something inside. Now what happens with the increasing temperature? Temperature increasing in tropical climate. So in tropical climate, all the plants used to photosynthesize as well as they used to respire. So with the increasing temperature, the CO2 concentration in the atmosphere decreases, and instead O2 concentration in the atmosphere increases. So in absence of lesser amount of CO2 and bigger amount of oxygen, the plant used to take oxygen more as compared to CO2. How it happens? It happens in this way. See, this RuBP, ribulose bisphosphate, it is a 5-carbon compound, and with the increase of oxygen concentration, the CO2 concentration depletes, and with the increase in oxygen concentration, O2 comes here. So there is no another carbon, because in CO2 there was one carbon. This 5-carbon compound splits. How? It completes with 3 carbon and a 2. That is, it itself splits into 2. How? Because this RuBP carboxylase acts as an RuBP oxygenase. It is known as RuBP oxygenase. So the same enzyme acts in 2 different ways. When there is concentration of CO2, it acts as RuBP carboxylase. When there is increase in oxygen, this same RuBP, rubisco, acts as oxygenase, and RuBP is split into 2. One is known as PGA. This is 3 carbon phosphoglycolic acid, and this is known as phosphoglycolate acid. Finally, RuBP is 5 carbon. 1, 2, 3, 4, 5. Ribulose bisphosphate or biphosphate or diphosphate. So here is one phosphate. Say here is one phosphate. We used to call ribulose 1,5-bisphosphate, or you can say ribulose 1,5-diphosphate. So it splits here. So 1, 2, 3, 1, 2, 3. Here is phosphate group. 1, 2. Here is one phosphate group. So this is known as phosphoglycolic acid. Let me write down the formula. This is phosphoglyceric acid and this is phosphoglycolic acid, 2 carbon compound. This is phosphoglycolic acid. So with the earlier it was considered the rate of respiration was same. Subsequently it was found that the rate of respiration in the light increases and in the dark it remains normal. Why? Because with the increase in temperature, with the increase in oxygen concentration in the atmosphere, so this RuBP carboxylase acts as an RuBP oxygenase and it splits the ribulose bisphosphate into a 3 carbon and 2 carbon compound. 3 carbon is known as PGA and this one is known as phosphoglycolic acid. Phosphoglycolic acid. Clear? It is having 2 carbon. Then this phosphoglyceric acid is immediately converted to glycolic acid. This is immediately converted to glycolic acid. So here P will go minus H3PO4 plus H2. It will be produced CH2OH COOH. This is the formula of glycolic acid. So CH2O hydrogen has come and phosphate has been removed. This is the acid COH group, carboxyl group is glycolic acid. What we say that with the increase in temperature and oxygen concentration, RuBP splits into phosphoglyceric acid PGA and phosphoglycolic acid. Phosphoglycolic acid then immediately converted to glycolic acid. Clear up to this? Now this glycolic acid reacts with peroxygens. Peroxygen present in the cell because where this RuBP and others are there in mesophyll cells. Peroxygen present in the mesophyll cells, it metabolizes the glycolic acid, it metabolizes the glycolic acid with an enzyme transaminase. With the help of enzyme the reaction is known as transaminase. That is, some amino group will come. Let me see. CH2NH2 COOH. Then it will be converted to CH2NH2 transaminase and COOH. This is known as glycolic acid and this is known as glycine. This is known as glycine. So, glycolic acid with the help of peroxygen in the cell metabolize the glycolic acid. The reaction is known as transaminase and some amino group is inserted. This is known as glycine. This is also an acid. COOH, it is acidic group acid. So mesothelin cell, see here what was going on and here something different is going on. Instead of production of this and that, one PGO of course it is produced, but this reacts from phosphoglycolic acid, phosphate group removes glycolic acid, then with the transformation amino group has increased, this is glycine. Now what happened to glycine? Glycine is immediately converted to serine, S-E-R-I-N-E, serine, with the liberation of CO2. With the liberation of CO2. The glycine immediately converted to serine with the liberation of CO2 with the help of, you write the name of this enzyme, serine hydroxy methyl transferase. Serine hydroxy methyl transferase. So glycine is then immediately converted to serine with the liberation of CO2 with the help of serine hydroxy methyl transferase. This is two molecules of serine will be here, CH2OH. Serine formula is CH2OH CONH2COH. This is serine. So two molecules of serine here, how it is coming from 2 to 3, that is two molecules of serine produced with the liberation of CO2. Our main target, you see, this is very important to note. You do not write the formula. This formula is just to understand. Simply you can write RuBP is converted to PGA and phosphoglycolic acid, and then phosphoglycolic acid is converted to glycolic acid. Then with the help of transaminase, with the help of peroxygen in the cell, it is converted to glycine. Glycine immediately converted to serine, and two molecules of serine is produced. And here some H2O, hydrogen peroxides, so many things comes. Do not write so many things. Is converted to serine with the liberation of CO2. This is the main factor, CO2. With the help of serine hydroxy methyl transferase. Why I am writing this CO2 in red? Because you can simply say some CO2 is going out, no problem. It is not like that. Because plant takes very much hardship to incorporate the carbon dioxide or especially the carbon. How? Because in the plant, in the leaf, in the chlorophyll, In the grana, photosystem 2 was there, photosystem 1 is there, and some photolysis of water and electron transport chain. So many things they have undergone to produce NADP and ATP. Okay, and then with the help of those ATP or NADP, this reaction took place. That is, Kelvin cycle took place. In the Kelvin cycle, plants also tried hard to incorporate the carbon. It is RuBP is the carbon acceptor. So RuBP reacts with 12. That is, it is not that at a time 6 CO2 is entering through the stomata. Single CO2 is entering and 6 times the cycle goes on. So plant is taking so many hardship to incorporate the carbon. And from this carbon they are to continue the cycle. Why? Because in absence of RuBP, no CO2 can be accepted. So he has to produce at least 6 molecules of RuBP. At the same time, they has to produce glucose. That is 1 glucose for 6 cycles. So they are taking hardship to produce carbon or to incorporate carbon. That carbon is liberated in case of temperature increase and O2 concentration increases. So they are losing carbon. It has been estimated the half of the carbon incorporated in the plant through a process of photosynthesis is lost by this liberation of CO2. That is, you should think, what goes on, and you should introduce or invent or something like that to prevent the plant from losing the carbon dioxide. This is known as photorespiration. That is the end. So I can conclude that you should write the first thing, this one, that this is the normal condition. When CO2 prevails in excess condition, this happens. And in the absence of CO2, oxygen concentration increases, and this happens with the liberation of saline and with the liberation of CO2. This is known as photorespiration. About half of the photosynthetically fixed carbon is lost by this photorespiration, which poses a threat to the plant, tropical plant, especially where temperature rises. This much is photorespiration. Thank you. Next time will come C4 pathway. C4 pathway, that is, plant itself has made some mechanism to prevent this oxidation of RuBP. Plant itself has made. I will come on the next. That is also a simple chapter. That is known as C4 pathway, beta carboxylase pathway, or Hash-Slack pathway. That will also take 5 to 10 minutes. Till then, thank you very much.